DNA, the blueprint of life. What do the following have in common? A strawberry, a tree, and a girl. DNA! Before you say it, I'm not talking about the studio album from Little Mix. DNA stands for deoxyribonucleic acid. It's the blueprint of life. In the same way that you have a blueprint for a house to help build it, DNA contains information to help build an array of organisms, such as dogs, birds, fish, insects, plants, and even microorganisms such as bacteria. We have known that DNA is a very essential molecule for life for a while now. However, its structure proved very difficult to solve, with a number of incorrect structures posed throughout history. It would have been easy to give up. However, in 1953, there was a scientific breakthrough as the scientific community had their eureka moment and finally solved the structure of DNA. The two scientists that are mainly credited for discovering the double helix structure of DNA are James Watson and Francis Crick. At the time, they were both at the University of Cambridge and collaborated with a physicist from King's College London, Maurice Wilkins. For their work, they all received a well-deserved Nobel Prize. There is an unsung hero in this story, and her name is Rosalind Franklin. She was a British chemist who helped come up with Photo 51, an X-ray diffraction pattern of DNA. Unfortunately, in 1958, she died aged 37 of ovarian cancer, possibly caused by the heavy exposure to X-rays. As a result of her death, she was unable to be nominated for the 1962 Nobel Prize in Chemistry. There are three key features of the double helix model. Two anti-parallel strands that twist around each other to form a helical structure. Four bases involving complementary pairing. A sugar phosphate chain that acts as the backbone for each strand. Unwound DNA looks like a ladder with the bases as the steps in the middle and the sugar phosphate backbone as the supporting poles. Note how the arrows are pointing in opposite directions to signify the anti-parallel strands. Let's make some DNA. Here are the ingredients: phosphate, deoxyribose sugar, and 4 nitrogen bases. The first step is to add a phosphate onto the deoxyribose sugar molecule. Repeat this step to form a sugar phosphate chain to form the backbone. Once the first backbone is formed, make the second backbone, ensuring that it runs in the opposite direction. Now for the heart of the recipe, complementary base pairing. Arginine pairs with thymine using 2 hydrogen bonds, while guanine pairs with cytosine using 3 hydrogen bonds. There are 2 types of bases, purines and pyrimidines. Guanine and adenine are examples of purines, and they are large bases, while thymine and cytosine are examples of pyrimidines and are small bases. There is another type of pyrimidine, uracil, but this is not seen in DNA. The way I used to remember this is to think of Tutankhamun and his pyramids. Tutankhamun was small when he died, so therefore the pyrimidines must be the small bases. Now that we've seen the structure of DNA, let's look at its function. DNA is mainly found in the nucleus of a cell. There is also mitochondrial DNA, which is inherited from our mother when we are born. Due to the length of DNA molecules, it has to be coiled up into a chromosome, which has 2 sister chromatids which are connected by a centromere. In each cell, there are 23 pairs of chromosomes, making 46 chromosomes altogether, with each parent donating 1 chromosome per pair. The last pair of chromosomes are slightly different, as these are sex-determining chromosomes. XY makes you a male, and XX means you're female. Fun fact: If you took all the chromosomes from all of your cells, you can stretch it from the Earth to the Sun 600 times. DNA contains a functional unit known as genes. Genes act like a computer software with a base sequence that encodes for a number of features such as hair and eye colour, which are known as phenotypes. We all have slightly different genetic base sequences, which makes us look all different. How do we get from the genotype, the genetic base sequence, to the phenotype, the physical, expressed gene products? The central dogma of cell biology states that DNA is transcribed onto a single strand of RNA molecule via a process known as transcription. The RNA molecule can then be converted into a number of gene products, such as keratin, which is found in hair, via another process known as translation. These two processes will need to be studied in more depth in a later video.